Good evening and good afternoon, Professor Murthy, Professor Dr. Hu Yu, and my dear colleagues and domestic and international hematology colleagues. This night we will have the joint meeting by Chinese Hematology Association and the International Association Clinical Hematology by the co-chairman. The chairman is the Chinese Hematology Association, Professor Hu Yu, and the chairman of the Professor Murthy by the International Association for the Clinical Hematology. So at first, we'd like to invite the Professor Hu Yu give us the open the regards of this joint meeting. Please, Professor Hu Yu.
Thank you for Professor Huang, distinguished Professor Murthy, Professor Huang, and colleagues. Good evening. It's my great pleasure to co-chair this China IACH Hematology Symposium with Professor Mohammad Murthy. On behalf of the Chinese Society of Hematology, I extend my warmest welcome to all distinguished guests, speakers, and participants from Europe and China to take part in this academic seminar from classical to innovative CAR-T. I'm delighted to announce that this symposium marks the first session of our series academic events between China and the IACH. In recent years, hematology has been advancing at an extraordinary speed, and cellular therapy has become one of the most promising fields, bringing new hope to patients with hematological malignancies. Facing rapid development and emerging challenges, close cooperation between top institutions across the countries is more important than ever. No single country or center can solve all problems alone. We need global hematologists to work hand in hand, share cutting-edge progress, exchanging clinical insights, and jointly promote scientific and clinical innovation. Today, we are here from distinguished speakers, including Professor Maria Victoria Matius, Professor Hen Mei, and Professor Yongxian Hu, followed by a panel discussion with outstanding colleagues from both countries. I'd like to express my sincere gratitude to Professor He Huang for his excellent organization and the coordination of this meaningful event. This platform not only fosters academic exchange, but also deepens friendship and mutual trust between Chinese and European colleagues. Finally, I wish this symposium a complete success. Thank you.
Thank you very much, Professor Hu. Thank you, Professor Huang. It is a great pleasure for us to organize this joint event, and a very warm welcome to all of you to this first ICH webinar in collaboration with the Chinese Society of Hematology. This is a milestone we hope will mark the beginning of a long and fruitful partnership. Today, we will explore the past, present, and future of CAR-T therapy, one of the most transformative advances in our field. And given the role of our Chinese friends and colleagues in the development of CAR-T cells, we believe this initiative truly reflects our shared vision of hematology without borders, where science, innovation, and patient care transcend geography. Thank you all for being with us, and we have prepared a beautiful program with Dr. Yishan Ye.
Yeah, thank you very much, Professor Hu Yu and Professor Modi. Very important opening remark. So I think this is a joint meeting. So in future we have some more topic about the leukemia, about the transplantation. So this night the first topic is cellular therapy. I think Professor Modi organizes IACH is a very popular advanced topic and pushes technology in the international clinical physicians. So Chinese Hematology Association also, they'd like to join the IACH and such is a serious lecture in futures. We will hold this one-for meeting and for the young doctor. So thank you for your both kind opening remark. So next we will enter to the lecture section. And the first topic is come from the Professor Victoria Mathies. Victoria Mathies is the consult physician in hematology and associate professor of the medicine in the University of Salamanca, Spain. She directed the myeloma program and coordinated the clinical trials unit at the Salamanca University Hospital. As the coordinator of the Spanish myeloma group, she has designed a lead key trial in end-to-end smoldering multiple myeloma, checking current treatment approach. She is the member of the IMWG and the EHA and the ASH. She has published over 140 peer-reviewed papers, serving as associate editor for myeloma in Annals of the Hematology. So today night, she will give us the topic about the cellular therapy and so give us the topic. So we will welcome Professor Mathies.
So first of all, thank you very much for the invitation to participate in this meeting. And of course, I have to confess that I am a bit overwhelmed talking about European CAR-T perspective in front of the Chinese hematologists that they have maybe much more experience than us. Well, I appreciate the invitation, and I will present to you the European CAR-T perspective. Here you can see my disclosures. All of you know well that myeloma is actually a very complex disease. And when we review the treatment overview, you know how after the discovery of the autologous and allogeneic stem cell transplantation, PI, IMiDs, anti-CD38 monoclonal antibodies, and the novel T-cell redirecting therapy emerged for the management of patients with multiple myeloma with a significant improvement in the overall survival. This was the treatment landscape for patients with multiple myeloma in Europe some years ago. And based on this slide, you can realize how after the first 2-3 lines of therapy, majority of the patients were triple class exposed, and the majority of these patients were triple class refractory. And we realized how this population was an unmet medical need, and this was evaluated in different observational studies, retrospective like Mammoth, or prospective like the locomotion, but well, we observed how once the patients were triple class refractory, there was not any new standard of care, and the outcome was actually very poor, with overall response rate approximately 30%, median progression-free survival approximately 4 months, and the median overall survival not superior to 1 year. And this was the rationale for the investigation on novel targets BCMA, GPRC5D, FcRH5, and others, and novel modalities of immunotherapy like antibody-drug conjugated bispecific monoclonal antibodies and CAR-Ts. And indeed, in Europe, this was the first experience we had with the CAR-T cell therapy. And indeed, today, ide-cel and cilta-cel are both approved in patients after three prior lines of therapy, so in 4-line plus, triple class exposed, and triple class refractory. ide-cel was indeed the first BCMA CAR-T approved by the European Medicines Agency. The rationale was 128 relapsed refractory myeloma patients. Median number of prior lines of therapy were 6, and 84% of the patients were triple class refractory. And in this situation, at the maximum dose, the overall response rate was 81%, the complete response or better 40%, median PFS 10.8 months, and median overall survival longer than 2 years. When we realized about this efficacy data, well, we said that this BCMA CAR-T covered the unmet medical need we had for the triple class exposed and refractory population. And this was the rationale for the first approval. But the cilta-cel was coming later on. And indeed, we observed how cilta-cel was first evaluated in the CARTITUDE-1 in 97 relapsed refractory myeloma patients, also after a median of 6 prior lines of therapy, majority of them triple class refractory. And cilta-cel showed even better results than those reported with ide-cel, because with cilta-cel in this situation, basically all patients responded, and over 80% of the patients achieved complete response. This is the last publication, and after a median follow-up of approximately 5 years, the median progression-free survival was 3 years, and the median overall survival 5 years. So indicating that this data clearly covered the unmet medical need, and indeed, in principle, cilta-cel showed to be more effective than ide-cel. Probably you know that in this cohort of patients, there was 1/3 of patients that remain alive and progression-free for more than 5 years, and 12 of these patients were treated at Mount Sinai Hospital in New York, and we are going to see later on, all of them were in minimal residual disease negative in the bone marrow, minimal residual disease negative by imaging techniques. It's true that in these patients, from the baseline characteristics point of view, no significant differences in comparison with those patients who had progressed within the first 5 years. The only difference is the plasma cell bone marrow infiltration, as well as the soluble levels of BCMA, lower in these patients that remain alive and progression-free for more than 5 years. But it's true that when the quantity and especially the quality of the T cells was evaluated pre-infusion and post-infusion, it is possible to see how at the moment of apheresis, the T cell neutrophils ratio was higher in those patients who remain alive and progression-free more than 5 years. And indeed, in these patients, in the drug product, the phenotype of the T cells was mainly naive for those long-term disease responders. When post-infusion, it was evaluated the effector target ratio. It was also higher in the CAR T cells for those longer responders, and the same is applicable to the CD4 CAR positives. The phenotype central memory was higher also in these patients. So maybe the biological explanation why these patients remain alive and progression-free more than 5 years is related with the quality of the T cells. As I previously told you, 12 out of these 33 patients were in MRD negative, sustained over time, all but one sensitivity level 10 to the -6, and these patients could be potentially cured according to the new definition recently defined in the workshop that took place in Miami in February. But also there are some other biological studies This is a complex study, but basically all these multi-omic analysis suggested that the durable responses is basically based on a balance between the immune recovery, basically characterized by the preservation of the CD4 memory T cells pools, as well as diverse and expanded T cell clonotypes. So maybe there are biological explanations in order to explain why this group of patients remain alive and progression-free. In terms of safety, you know maybe better than I, but CRS with ide-cel and cilta-cel is something expected. I would say that it is predictable and manageable in majority of the patients. In terms of ICANS, in these two clinical studies conducted in heavily pretreated myeloma patients, ICANS is not very different with ide-cel and cilta-cel, between 16 and 18% of the patients. But as you know, the problem is another neurotoxicity that has been reported with the cilta-cel originally in the CARTITUDE-1, and this has been confirmed in subsequent clinical trials and the real life. And I do refer to the Parkinson's syndromes, as well as the cranial nerve palsy, other peripheral neuropathy, enterocolitis, and why not to refer to the second primary malignancies that personally I consider that in the CARTITUDE-1, this is not a relevant issue because these patients were actually heavily pretreated, and these patients would have died if they would have not had the possibility for being treated in this case with the cilta-cel. Cytopenia and infections are comparable with both CAR-Ts, ide-cel and cilta-cel, and we have to try to protect our patients from infections with the adequate prophylaxis in immunoglobulins at the beginning, as well as the adequate vaccinations. In terms of quality of life, in both clinical studies, CARTITUDE-1 as well as the KarMMa trial, patients experienced a very positive experience from the quality of life, basically because majority of the patients responded and they remain without treatment for some months, definitely more months after cilta-cel than after ide-cel, but overall there is a benefit in quality of life. This is the situation in the US. This is not in Europe because we don't have this information yet. But you see how at the beginning ide-cel was used, and as soon as cilta-cel was approved, there was a peak in the use of cilta-cel. And today, I think that this is also applicable to Europe. Cilta-cel is more frequently used than ide-cel. Head-to-head comparison was done in this retrospective study in the real life, and it's true that cilta-cel seems to be better in terms of PFS and overall survival, and in terms of non-relapse mortality, no significant differences between cilta-cel and ide-cel. Any other CAR-T-cell therapy for triple class exposed that they could potentially be incorporated in the clinical practice in the future here in Europe? Well, we have this academic CAR-T. This is applicable to Spain, but as I had the opportunity to share with you this slide, probably you know Cisnacel is a BCMA CAR-T that we have here in our country. It was evaluated in heavily pretreated myeloma patients, and the outcome is quite acceptable. The median progression-free survival is around 1.5 years. Safety profile is quite good. And from my personal perspective, I think that the possibility for having academic CAR-T increases the flexibility, and the possibility of a point-of-care approach clearly facilitates a lot the possibility for prescribing CAR-T to some specific patients. Anito-cel is another BCMA CAR-T. It is not approved yet in Europe. We expect this BCMA CAR-T to be approved in the US at the end of this year, and we will have maybe the approval in Europe based on this phase 1 study, or we will have to wait to the phase 3 clinical study. This BCMA CAR-T presents these D-domain attributes, that this is a non-antibody-derived synthetic protein that can increase the efficacy in comparison with other BCMA CAR-Ts because facilitates high transduction efficiency, CAR positivity, and CAR density on the T-cell surface. In addition, the size is smaller in comparison with other CAR-Ts, and this can facilitate the penetration in the extramedullary disease. These are the data we have coming from the phase 2. This is the registration study in the US, and basically the efficacy, I would say that it is comparable to cilta-cel, and from the safety profile point of view, it is quite acceptable, and no delay or non-ICANS neurotoxicities have been so far reported, no cranial nerve palsy, no Guillain-Barré, no enterocolitis. So in principle, the toxicity profile is predictable in comparison with what we've seen with cilta-cel. Arlo-cel is another CAR-T that it is ongoing now also in the US, GPRC5D CAR-T. And this CAR-T is important because it was evaluated in relapsed refractory myeloma patients, but 46% of the patients had been previously exposed to BCMA-targeted therapy. And overall, the overall response rate is quite encouraging with almost 90%, 50% of complete response or better. This efficacy is maintained in the population previously exposed to BCMA therapy, what I consider that it is relevant. And we have other CAR-T that it was first evaluated in China, now it is under evaluation in the US, and we will have this in Europe. This is the reason why I decided to present here. Dual CAR-T targeting BCMA and CD19. And again, some patients had been previously exposed to BCMA CAR-T. You have to know that in Europe, this is the new unmet medical need we have. The population exposed already to either ide-cel or mainly cilta-cel, and how to rescue these patients with other potential CAR-Ts. And in this case, the dual CAR-T showed to be equally effective. And from the safety profile, I would say that the CRS, ICANS, infection, cytopenias, but again, no delayed neurotoxicity, no enterocolitis, so no toxicity that I previously reported for cilta-cel. But according to the International Myeloma Working Group consensus guidelines, according to the European Myeloma Network consensus guidelines, well, the idea is to move with the BCMA CAR-Ts earlier on, basically because the T cells are going to be more functional, because the fitness of the patient is going to be better, and because the disease will be less aggressive. This is the reason why we have ide-cel evaluated in the KarMMa-3 in patients after 2 to 4 prior therapies. I have to let you know that ide-cel in earlier lines of therapy was approved by the European Medicines Agency, but basically not reimbursed across the different European countries. In this study, ide-cel was evaluated against different standard regimes, and in spite of the fact that the median number of prior lines of therapy was 3 and the population was less heavily pretreated, 65% of the patients were triple-class refractory, and benefit in PFS, but the median PFS was 14 months for ide-cel, no benefit in overall survival. And this is basically the reason why ide-cel in earlier lines of therapy has not been reimbursed in the majority of European countries. We have, however, approved in Europe cilta-cel after 1 prior line of therapy. The only criteria is the patients had to be exposed to PI and IMiD and refractory to lenalidomide. Cilta-cel was compared with either PVD or DPD, and you know that this is the first BCMA CAR-T showing a significant benefit in progression-free survival with a hazard ratio of 0.29 and also benefiting overall survival with a hazard ratio of 0.55. This benefit is maybe derived from the efficacy In terms of minimal residual disease negativity rate, in the evaluable population, almost 90% of the patients reached MRD negative 10 to the minus 5, 85% 10 to the minus 6. And here on the right-hand side of the slide, you can see how the median time to MRD negativity was quite rapid, but some patients at the same time had the possibility for upgrading the quality of the response over time. And again, when we compared the quality of the T cells at the apheresis as well as at the moment of the infusion in those patients in MRD negative versus those patients in MRD positive, as expected, the proportion of patients at apheresis CD4 T cells with a phenotype naive impacted in the probability for achieving MRD negative. By contrast, when the product at the moment of the apheresis contain, well, many patients, many lymphocytes with a phenotype effector memory, this negatively impact in the probability of achieving MRD negative. But at the moment of the pre-infusion, interferon gamma, so inflammatory marker, also negatively predicted MRD negativity rate. Soluble levels of BCMA at pre-infusion didn't impact in the outcome, but the peak of expansion clearly impacted in the probability for achieving minimal residual disease negative. And minimal residual disease negative, we know that it is an excellent surrogate marker for outcomes, and in those in MRD negative at 1 year, so 93% of the patients remain alive and progression-free at 30 months, and 97% of the patients remain alive. But as I previously said, other CAR-Ts that will be incorporated into the European market in the near future will be anito-cel, based on this phase 3 clinical study. This study has already completed the recruitment, and we are awaiting for the results in the upcoming years. The same is applicable to Orla-cel, the GPRC5D CAR-T. The quintessential 2 phase 3 clinical study is ongoing, and it is comparing Orla-cel with either DPD or KD. So this is a registration study, and we will have in Europe in the future this CAR-T maybe, and the same is applicable to the BCMA CD19 CAR-T developed in the DURVA-4 clinical study. This is also a phase 3 clinical study in relapsed refractory myeloma patients evaluating this BCMA CD19 CAR-T against different investigator choices standard of care. This means that, well, today this is what we have in Europe, and today CAR-T cell therapy is only available in the relapse outside of clinical trials, but in the future, I would say that we will have maybe from second line, not only cilta-cel, but as I previously said, anito-cel and durva-cel, and why not also Orla-cel targeting GPRC5D. In first line, we have to wait. We have a CARTITUDE-5 and CARTITUDE-6, and these clinical studies already completed their recruitment. You know that CARTITUDE-6 is comparing cilta-cel with the transplant. In the CARTITUDE-5, single infusion of cilta-cel is being compared with VRD followed by Lendex in the transplant ineligible population, but other trials are ongoing. This is a clinical trial that we are conducting in Spain with anito-cel in first line. This is, well, these are actually 3 cohorts of 10 patients each, 1 patient transplant eligible, 2 transplant ineligible, and we are developing this clinical study, and this is going to be the rationale for a phase 3 clinical study, maybe in the transplant ineligible as well as in the transplant eligible population with anito-cel. What about high-risk smoldering? Again, although this study was conducted in the US, I decided to show you here because this study has just been published and presented in the AAC meeting by Irene Gabriel last week. Cilta-cel in 20 patients with high-risk smoldering. and different dose modifications were done throughout the study in order to improve the safety profile. From the efficacy, no problem. All patients in MRD negative, 100% of the patients alive and progression-free. And from the safety profile, it's true that 7 patients develop neurological toxicity non-ALCANs, including facial nerve palsy, peripheral neuropathy, tremor, and movement and neurocognitive disorders. It's true that all of them were grade 1. There are 3 patients in which these abnormalities are ongoing, although improving. And this study evaluated cilta-cel in high-risk smoldering myeloma without any bridging therapy. And we are going to develop here in Spain, and indeed we have already started the recruitment of another 20 patients with high-risk smoldering with cilta-cel, but at dose of 0.3, and all patients are going to receive 2 cycles of Dara-BRD in order to reduce the tumor burden. So mechanism of resistance after CAR-T, I am not going to discuss about this, but of course we have to take them into consideration in order to see how we are going to proceed after CAR-T, what is the appropriate rescue therapy, bispecific monoclonal antibodies, or maybe other CAR-Ts. And I finalize with this, and in Europe today we have ide-cel and cilta-cel in relapsed refractory myeloma, but as I showed you in my presentation, other CAR-Ts will be incorporated in the clinical practice once the phase 3 clinical studies that are ongoing are positive, and the next step is to move with these CAR-Ts into the first line of therapy. And I stop here. Thank you very much for your attention.
Okay, thank you very much, Professor Maria Victoria Matos, and it's a wonderful, nice talk about this perspective CAR-T cell in the European. So I think we have the total 3 lecture. After the lecture, we have the together discussion. Okay?
Perfect.
Yeah, thank you so much. So next talk is the Professor Meihen. Topic is the progression in the clinical translation research of the in vivo CAR-T. Dr. Meihen is the director of the Department of Hematology and the Union Hospital, Tongji Medical College, Huazhong University of Science and Technology. He is published the first clinical report about in vivo CAR-T for multiple myeloma. I think it is a very important article about the CAR-T advances technology. He is the recipient of the National Science Funding, a distinguished young scholar, and the research focus on the precision medicine in the hematology disease, and lead over 10 major national research programs, and including the KING program from the National Nature Science Foundation, and published about the more than 70 SI paper, and as the first of the corresponding author. So today he will give us the talk about in vivo CAR-T. So welcome, Professor Meihen.
So it's my great pleasure today. So I'll go ahead to get the time up. My topic today is the in vivo CAR-T therapy for relapsed and refractory multiple myeloma. So as we all know, CAR-T has demonstrated a remarkable efficacy, such as Emily remains cancer-free for more than 30 years since FDA approved the first CAR-T commercial product, Kymriah, in 2017. And now 15 products have come to the market. I think there are 9 products in China now, also contain 4 types of CAR-T products for multiple myeloma. So looking back at the development of CAR-T for multiple myeloma, just as the professor mentioned for the CARTITUDE study, from the NEGINA study in 2016, and then it's the CARTITUDE in 2018, and to CARTITUDE 6 and KarMMa-9, also the GC12 and the GPRC5 and the FCIH5. So the CAR-T in myeloma is doing from deep responses to long-term survival, and later to the earlier lines of therapy, and from the complex manufacturing to faster production availability, and also from the classic BCMA single target to BCMA plus CD19, BCMA plus GPRC5D, BCMA plus CD8, novel dual targets. However, conventional CAR-T still has some limitations. The ex vivo CAR-T manufacturing process is very expensive and complicated. According to the statistics, about 1/4 patients died when they wait for commercial cilta-cel in the United States. So the accessibility is a major use in the clinical application of CAR-T for multiple myeloma patients. And moreover, conventional CAR-T exhibit poor infiltration and limited efficacy in solid and extraordinary tumors. Novel technology to overcome the limitations of autologous CAR-T, that is in vivo CAR-T. So the T cells are directly edited in vivo into CAR-T cells within the patient's body using viral vector or nanocarrier, establishing a new paradigm for the CAR-T therapy. So we published a review for compare with autologous and universal CAR-T. The in vivo CAR-T is not required to the ex vivo constitutionalization, and it generates the cells is from the in vivo, not only for T cells, but in the future, maybe NK cells, macrophages can be repacked in vivo. And also, there's in the clinical processing, there's no need for the lymphodepletion pre-treatment for this patient. And the best advantage for that is the large batch of yield of the in vivo CAR-T, maybe up to 1,000 units per batch. So the cost in the future, the CAR-T will be very cheap. So the accessibility also the in vivo CAR-T is the best. And there are different strategies for how to generate in vivo CAR-T. And the most important thing is the carrier. So most now common is the 3 directions. So one is lentivirus, and another one is the AAV, and also LNP. So compared with the traditional gene therapy or cell therapy, the differences for the in vivo CAR-T, I think they merge them together. No matter viral vectors or nanocarriers, they are only for the carrier, but they need to also the T cell binder is very important. So normally there are CD3 or CD7, CD5 TCR can be used as the T cell specific surface molecules. And another thing is the expression is very important. So LV is the integrates into the genome. So this is the advantage for that, but also that is the side effect for that. But for AAV and LNP, there's no integrating into the genes, so it could be safer than LV. But the efficacy for the LV is higher than AAV and LNP. So the safety is not easy to say now, but also the cargo is also very important for the delivery system. So since 2017, the first concept for the in vivo CAR-T is developing very quickly, especially after 2024. Our team firstly implemented into in vivo CAR-T treatment for multiple myeloma in November 2024 and published the first human beings data in the Lancet. Later in the ASH meeting, the group from the Peter Mac released their data of Canolia 1010. Here I will present the detailed data from these studies. First is the ESO-T01 by our team, ESO-Bio and Pregene. ESO-T01 is a 3rd generation and no replicating self-inactivation lenti-vector containing the anti-TCR VHH for binding to T cells. So it is different than other CD3 or CD7. And mutated VSAG for envelope. And CD47 to reduce vector phagocytosis. And MHC1 knockout to reduce the immunogenicity. And we also BCMA CAR and T cell specific promoter made it more accurate and safer. So in the cell line assays, ESO-T01 effectively lyses target cells, outperforming the positive control CD cell at no effector to target ratios. And in the humanized mouse study, ESO-T01 demonstrated robust anti-tumor efficacy across all dose groups. The lowest dosage is 7 × 10⁵ TU. The administration prolonged the survival time in the mice study. There has no significant differences change in mouse body weight and no notable clinical abnormalities for this observed mouse animal. We found that the transduction is most to T cells, but not big difference between CD4 or CD8 positive T cells. And also the biodistribution is in the liver and in the spleen and in the bone marrow of the humanized mouse. Very importantly, the vector biodistribution assessed by VCN assays confirm that although the vaccine can be divided into the liver and the spleen, but only for the spleen there are expressed BCMA CAR T cells. So that confirmed maybe the in vivo CAR T cells was generated in the mouse spleen. So I think this might answer how CAR T is produced in the body. So here is the dose selection is based on the safe and effective dose in humanized mouse, as well as those from similar products in non-human primate studies, such as AAV studies. The minimal effective dose in the humanized mice model is 0.7 × 10⁶ TU. For safety consideration, the clinical starting dose is typically 1/10 of the effective doses in animal model. So use 0.2 × 10⁹ TU per subject of the ESO-T01 injection as the starting dosage. So here is the inclusion criteria. It's very familiar with all the experts for melanoma study. We emphasized received at least 2 lines of anti-melanoma therapy, including both immunomodulators and proteasome inhibitors. And we very importantly, we emphasized the absolute the CD3 positive T cells should be above 150 per milliliters. But now for the studies we are carrying out, we make this more higher. I think will be better for the product of the generated T cells. And here are the exclusion criteria. So what patients should exclude the patient targeted therapy within 5 half-lives, and what patient should not have VSVG antibody positive. And the first 4 patients we enrolled were all those who had relapsed after undergoing high-intensity treatment on the front line, including sequential autotransplantation and CAR T treatment. There are 2 patients, and 3 of them have cerebrospinal fluid involvement, and all of them have extramedullary disease, and the largest measurable lesions is above 11 centimeters. I think for these 4 patients, all will be excluded for traditional CAR T clinical trial or commercial products. So it's very hard to treat. And we observed the 2-phase CRS phenomenon in all 4 patients after reinfusion, especially for the first patient suffered with very high CRS. You can see the IL-6 is very high, and the disturbance of the carcinogenesis, and the IL-6 level is very high. So it may be related to the acute inflammation reaction induced by the vector. So I'm not sure how the exact mechanism behind, but I call it that's vector induced. So with the high dose of dexamethasone for prophylaxis from the second patient. So from the second patient to 4 patients is much safer than the first patients. The adverse reaction have been significantly reduced. And we monitor the various laboratory indicators, including IL-10 and fibratin, and also CRP and some liver function index such as LT-AST and also GGT. And all these index exhibit biomodel distribution. And the viral RNA was detected only in peripheral blood sample within 48 hours. So I think after that, will be much safer than in the first 2 days. And everybody think about if the in vivo CAR-T is the side effects is heavier than the traditional CAR-T. But when we monitor the hematological toxic assay, we found that maybe there's a similar to that. And even for the hematological index, the in vivo CAR-T I think is safer than that. But why for that? Because you can see for the hemoglobin concentration is very stable at the front or after the experiment is very stable. And also for platelet count, and even after 15 days, it rises very quickly. But for our BCMA treated patients, for the traditional CAR-T, it always result very long time of for some high-grade CRC patient, it will last very long time for the decrease of platelet count, and even call it ICHT now. But for the in vivo CAR-T, it's very stable. And only the decrease is the blood count, is the white blood cell count, and especially for the sharply decrease of the lymphocyte count. But now I think the sharp decrease for this is very important. It shows how this in vivo product work. If there's no this sharp decrease, maybe it's not a good in vivo CAR-T product. But if virus infected, such as COVID-19 in Wuhan in 2019, some patient is suffered very heavily, they will not recover again. But for in vivo CAR-T, it's different from that. For this patient, will recover very quickly. In 2 days, it will recover very again, and the lymphocyte will rise again. So then I think that is very different from the traditional. So here there are some follow-up data for our 4 patients. Regarding to the results, the best response for these 4 patients are 2 SCR and 2 PR. The first patient get SCR but relapsed at 4 months and resumed to receive chemotherapy, but failed to achieve a response and died of disease progression at 7 months. And the third patient relapsed, got a PR at first and relapsed at 3 months and resumed to receive ex vivo BCMA GPRC5D CAR-T treatment. Because when we test, when he relapsed, we found that the expression of BCMA is lower than before, about 50% of that. So then we think there may be the immunological resistance for that. So we choose the dual CAR-T for that again. And very interesting, then she achieved the VGPR again at month 1 post-infusion. But unfortunately, she died because of the intracranial hemorrhage at 6 months, because for this patient, she suffered with 2 times of the CAR-T treatment. So the blood count, especially platelet count, is very low. And for the fourth patient, he got a PR at first and relapsed at 3 months with MRD positive and CSF infiltration, and withdrew to chemotherapy again, and achieved SCR again at 7 months, and died of disease progress at early this January. So it's totally 10 months alive. So now we still have 1 patient who got SCR at the first 28 days and is continuous CR. So till now it's more than 50 months now. So I think this is the longest time for the patient treated with in vivo CAR-T in the world. So here are some changes in the extradominary lesions, and I think this is the most amazing result for me. So you can see not matter the left side of the sternum and also right nipple and also the right pubic region. And this is, I think this is most difficult for the lesions in the muscle. So you can see after 10 days, the muscle, the legend is half of that. And after 28 days, it is discrete. It is what we regard as for these lesions is very interesting for this patient. And here the PET-CT also showed a significant regression of the systematical tumors. That is the before, and this is after for different patient. And we can also test the pseudo-BCMA as the marker for that. And the pseudo-BCMA becomes lower for this patient. And we also compare the kinetics of the in vivo BCMA CAR-T with the commercialized BCMA CAR-T product. So I think this is very important because and when we submit this data to the editor, they like that very much because it displayed a very good expansion, especially on the Cmax and the OCT28. So we got this data from the FDA website, and we compared it for the EASL TO1 and also IDCell. So we can see the Cmax is very important for a CAR-T product. If it is very high, it means the response rate will be very high. So the CR rate and also MRD CR rate will be high. So that's maybe why for this patient, the maintenance of the CAR-T cell is not very long, only for maybe for 2 months and disappeared in 3 months. But it still have very good result because the expansion is very important, I think. And another very important test that is we compare with the different places of the CAR-T infiltration. Because for in vivo CAR-T, the best advantage for that is maybe it can generate the CAR-T cells in the original site of these tumors. So we tested this for the tumor tissues. We found that there are about 31.7% CAR-T cells in the tumor tissues, and before infusion and the peak expansion. And here is the IHC showed that the tumor also markers significantly regressed in these patients. And also we confirmed this by qPCR. It also very high expression for these tissues. And this also can be detected for the CSF and also for the preliminary infusion, or these tissues have a very high amount of expression of these CAR-T cells. And here we also monitor the phenotype of different CAR-T cells and the peak expansion in the human beings. So here we can see the EASL TO1 only transduced to T cells in human bodies. So this is very similar to the result in the humanized model. And patient 1 and 3 exhibit a high promotion of the CD8 T cells, with patient 2 and 4 showed a dominance of CD4 positive T cells, but limited because the limited cases, so we cannot exclude if 34 positive or CD8 positive is better, or the ratio should be better. And except for the patient 3, all cases were primarily dominated by live and central memory phenotypes. So that is the patient 3. You can see for the effective immune cells is very high. So it's a significant promotion of CD28 negative, but CD57 positive syncytial T cells. So which correlated with the poor CAR-T expansion, I think. So, and we also compared different CD3 negative cells or NK cells. So all of them have no high expression of these CAR cells. So this is our preliminary data. So from now on, in the world, there are 3 groups. One is from our hospital, another one is from Tongji Hospital, and another is from Peter Matt. We will use the BCMA targeted in vivo CAR-T to prove the concept of in vivo CAR-T in human beings. But I think the most advantage for this, that is for in vivo CAR-T, is ready to use, and it may have a low cost in the future and a high accessibility for this patient. But the challenge is that there is still unknown safety, and even some people died in the center, in other centers. And the targeting specificity is still should be better. And how to avoid immunogenicity is still a problem. But in the future, I think the area of in vivo CAR-T has arrived. From the Nature Science, the National Development Journal, world papers have led the trend tendency of the in vivo CAR-T in the future. So the powerful cancer-fighting cells will soon be cheaper and quicker to make. So here my presentation today. Thanks to the colleagues in my team, and thanks to the patients enrolled in our clinical trials, and thanks to collaborative companies.
Yeah, thank you. Thank you very much, Dr. Mei. It's a wonderful talk about the in vivo CAR-T. I think it is very important data and the milestone in CAR-T therapy in multiple myeloma. So we will discuss together after the lecture. And next speaker is Professor Hu Yongxian. I'd like to introduce Professor Hu Yongxian. He's the Vice Dean of the Zhejiang University School of Medicine and recipient of the National Science Fund for Distinguished Young Scholar. She is Zhejiang University Distinguished Professor and the Deputy Director of the Bone Marrow Transplantation Center and the first ever hospital, Zhejiang University School of Medicine. Her research focuses on cellular therapy and published so many high impact papers as first or the corresponding also, including the New England Journal of Medicine, Nature, and the JAMA Oncology. And so today, she gives us a talk about the CD7 CAR-T therapy for hematological malignancies. Please, Professor Hu.
Thanks for Professor Huang's introduction. Dear Professor Monty, Professor Hu and Huang, and distinguished experts and colleagues, it's my great honor to have this lecture. My lecture is CD7 targeted CAR-T therapy for hematological malignancies. There are three challenges for the CD7 CAR-T therapy. First is the fratricide of the CAR-T cells because both CAR-T cells and leukemia cells express CD7. So for these CAR-T cells, they will kill themselves, each other. And the second one is the T-cell aplasia because the CAR-T cells will also kill the normal T cells because of the CD7 expression. And the third one is the contamination of the tumor cells because the tumor cells are CD7 positive tumor cells and the CD7 positive. So when we collect the T lymphocytes, these T cell tumor cells will also be collected to make CAR product. So the contamination of the tumor cells in CAR-T product is also a very important question. According to this, we think there are high risks for the autologous CAR-T cells for these patients with T-cell malignancies. Maybe we would prefer to the donor-derived or universal CD7 CAR-T cells for these CD7 positive T-cell acute lymphocytic leukemia or lymphoma. So my topic includes 4 parts. First one, sibling donor-derived CD7 CAR-T for T-cell acute lymphocytic leukemia, lymphoma, or acute myeloid leukemia. And the second one is universal CD7 CAR-T cells for T-cell acute lymphocytic leukemia and or lymphoma, because these universal CAR-T cells are derived from unrelated donors. They are totally HLA mismatched. And the third one is integration of CD7 CAR-T therapy and haploidentical stem cell transplantation. And the fourth one, we found that there are some special complications after CD7 CAR-T treatment. So first we have established these sibling donor-derived CD7 CAR-T cells. And these CAR-T cells are mostly HLA haploidentical or totally matched sibling donor. And most of the donors are haploidentical donors. We use these CD7, we use the intra-block technology to prevent the CD7 cell surface expression to avoid this fragile site. So from this figure, we can see these CD7 CAR-T CD7 are not expressed on the CAR-T cells. And also we found after CD7 block is intracytosol, not in the surface expression. These CAR-T cells also exhibited significant anti-tumor activity, both in vivo and in vitro and in vivo against CD7 positive malignancies. Then we have our IIT clinical trial. The first one is for the T-cell acute lymphocytic leukemia and lymphoma. this is a phase 1 dose escalating clinical trial. We just screened for 18 patients and 5 patients were enrolled. First 3 patients were infused with the low dose CD7 CAR T cells, about 2 million per kilogram. And the second dose is 4 million per kilogram. And then we expanded this dose for 9 patients. And we can see the efficacy, 14 patients were evaluated for the efficacy analysis. And we found 14 patients achieved 15 patients for the efficacy analysis and 14 patients achieved CR or CRI. So 8 patients bridged for allogeneic stem cell transplantation, but 2 patients died of relapse and 2 patients died of the infection and 1 died of the hemorrhage and 1 died of the EBV associated lymphoproliferative disease and 1 for persistent CR. And 6 patients without further treatment after CAR T therapy. 5 patients died of relapse and 1 died of EBV associated lymphoproliferative disease. So we think the CD7 CAR T cells can eradicate most of the CD7 positive leukemia, but maybe how to maintain long-term remission is also a very important question. This is the side effect analysis. We found the pancytopenia is also an important, very big challenge. Maybe more than half of the patients have suffered from pancytopenia more than 1 month. Another one is the virus infection, especially for this EBV virus infection. And then we have another clinical trial about relapsed refractory acute myeloid leukemia. These patients should have CD7 positive in the acute myeloid leukemia cells. And we screened 21 patients. Finally, 14 patients were enrolled for this clinical trial and infused the CAR T cells. Also, 6 patients received the dose escalation, and 8 patients was divided to the dose expansion cohort. For this trial, we also found that all the patients, if they have CD7 positive leukemia cells, so these CD7 CAR T cells can all eradicate and can totally eradicate this CD7 positive acute myeloid leukemia. But for those cannot reach CR or relapse, there are all CD7 negative acute myeloid leukemia cells. Even after transplantation, some patients will relapse with CD7 negative acute myeloid leukemia cells. This is a safety analysis. We found that the CRS is not very severe. 13 patients have suffered from CRS, and 13 with grade 1 or 2, and 1 with grade 3. And for the ICANS, only 1 patient had grade 1 ICANS. And we used the tocilizumab plus low-dose glucocorticoid in 2 patients and low-dose glucocorticoid in 3 patients. So all the CRS and ICANS can control very well. Our second part is universal CD7 CAR T cells for the T-cell acute lymphocytic leukemia and lymphoma. This is from the unrelated donor. So because the donor CAR T and the recipient T cells, they are HLA type are totally mismatched. So we should knock out the TCR in the CAR T cells to avoid GVHD. We also knock out CD7 to avoid the fratricide. And because of the CD7 and TCR knockout, so we knock in the gamma chain to rescue the interleukin signal. and also to avoid the host immune cells to attack the CAR T cells. We also knock out the HLA-2 in the CAR T cells and knock in the NK inhibitor to reduce the rejection by NK cells. For this multiple gene editing, we use the CRISPR-Cas9 gene editing. So this is efficacy. A total of 11 patients were included in the efficacy analysis. None of these patients reached overall response. Of the 8 patients with leukemia, 6 patients reached CR or CRi, and 3 patients with lymphoma responded to treatment, and 1 with CR. 3 patients with leukemia and 1 patient with lymphoma experienced disease progression. For these patients, 6 developed EBV-associated diffuse large B-cell lymphoma after reaching CR and died on 93 days after infusion. And 3 patients were bridged to allogeneic stem cell transplantation 3 months after infusion and were in continuous remission. So this is the kinetics of these universal CAR T cells. We found the CAR T cells reached the peak expansion on 10 to 14 days after infusion. Also, for this qPCR, we can detect the CAR T cell expansion. But we found that after CAR T cell expansion, CD7 positive T cells were disappeared, and CD7 negative normal T cells just compensate for these CD7 positive T cells. Then we found that the CD7 negative T cells are mostly CD7 positive. And these CD7 positive, CD8 positive, CD7 negative T cells are just the recipient derived. So as these T cell subsets expanded, they just eradicate the universal CAR T cells. So this can explain why these universal CAR T cells cannot persist for a long time in the recipient. So we are trying to do our next generation CD7 CAR T cells, just how to prevent these CD7 positive, CD8 positive, CD7 negative T cells to reject the universal CAR T cells. And I will introduce my third part, integration of CD7 CAR T cells and haploidentical transplantation. So from our previous clinical trial, we found that there are 3 challenges. First is pancytopenia of the CAR T treatment. And the second is relapse is also a major problem. And the third is EBV activation. So how to deal with these challenges? This is the law for a myeloablative high-dose chemotherapy for our conventional allogeneic stem cell transplantation. Maybe first, this chemotherapy will eradicate residual leukemia cells. And second, the chemotherapy can also inhibit recipient immunity to promote donor cell engraftment. And the third, after this high-dose chemotherapy, it will provide donor hematopoietic stem cells niche for the niche. And from our clinical trial, we found that after CD7 CAR T cells, the CD7 CAR T cells eradicate all the leukemia cells because we found that after CD7 CAR T treatment, the bone marrow cannot detect any leukemia cells, or even the extramedullary site also can be eradicated. And the second is the CD7 CAR T cell eradicated recipient derived CD7 positive T and NK cells. After bone marrow examination, we found these patients after CD7 CAR T, their bone marrow are aplasia. And we also found that the CD7 CAR T cells eradicate donor-derived CD7 positive T cells that will cause GVHD. because both allograft, these CD7 positive cells were all depleted by these CD7 CAR-T cells. And even after the CD7 T cells differentiated from CD34 positive stem cells, these T cells were all CD7 negative. So our protocol, we just have our new protocol. We just think the allogeneic stem cell transplantation after CD7 CAR-T cells without myeloablative chemotherapy and GVHD prevention. So this is our novel transplantation strategy. Just after CD7 CAR-T therapy, when these patients are in pancytopenia, we just transfuse the donor hematopoietic stem cells. And for this strategy, just CD7 CAR-T and donor stem cell are from the same donor. We just do not need the myeloablative chemotherapy and immunosuppression for GVHD prophylaxis. So this is our, we call the all-in-one strategy that integrate CD7 CAR-T and hypoidentical stem cell transplantation. We will enhance this lymphodepletion chemotherapy because the CAR-T cells are derived from hypoidentical donors. So we use the fludarabine CTX and we P16 for five days, just maybe a more severe than conventional FC lymphodepletion chemotherapy. Then we will transfuse the CAR-T cells. Then these patients, the CAR-T cells will expanded and eradicate the leukemia cells. After the CRS finished, we will evaluate the patients. If the patient, just for the bone marrow examination and blood test, if the patients are in CRI with severe bone marrow hypercellularity, and at the same time, these patients should have detectable CD7 CAR-T cells, then we will have these patients selected for allogeneic stem cell transplantation, just give them the same donors hematopoietic stem cells. So these patients do not need a pharmacological conditioning chemotherapy and also no GVHD prophylaxis drugs. We think for such all-in-one strategy, there are four advantages. First, free of pharmacological conditioning. So even if these patients have insufficient function of the organs, they can also tolerate this strategy. The second, free of the pharmacological GVHD prophylaxis, and this will also enhance the host's immunity. Also, because of no pharmacological conditioning regimen and no pharmacological GVHD prophylaxis, so the CD7 CAR-T cells will persistent in the recipients just for a long time. So even after transplantation, these patients still have high levels of the CD7 CAR-T cells. And the fourth one is that because the donors of the recipient, in the recipients, the immune cells will also derived from the donor stem cells. So these patients will still have the graft versus leukemia potential because of the allogeneic HLA disparity. This is our 10 patients efficacy. The median follow-up duration for the survivors was 15 months. And the longest one was 24 months. As of the cutoff date, 6 patients just remained in MRD negative CR without any treatment. So these patients have a very high quality, life quality, I think. We also detected the T cell dynamics, and we found these CD7 positive T cells were completely eradicated at a median time of 8.5 days, accompanied by expansion of the CAR negative, CD7 negative T cells. So these T cells also have immunity, but they can very weakly induce the GVHD. Also, we found these CD7 positive T and NK cells remained undetectable until the last follow-up. So all the patients are reconstituted with CD7 negative T cells. So after transplantation, we also found challenges exist. First is CD7 negative relapse, and the second is EBV positive lymphoproliferative disease. So this is our fourth part, CD7 CAR T cells associated complications. For this is from our first 10 patients. We found 2 patients with acute myeloid leukemia relapsed with CD7 negative. All are 2 acute myeloid leukemia patients. We can see this is before CAR T treatment, and the patient have maybe strong CD7 expression, and this patient achieved a complete remission after CD7 CAR T and the transplantation, but finally she relapsed with CD7 negative. And this patient is the same after CAR T and transplantation relapsed with CD7 negative. So we just collected all the CD7 negative relapsed patients as the leukemia samples. We just have also the CD7 positive leukemia cells before CAR T treatment, and also have the CD7 negative leukemia cells after CAR T treatment. So we use this WES and found that CD7 in the exon CD7 of the CD7 positive poly-CAR T and relapsed CD7 negative tumor cells, acquired flank shift mutation was detected in the exon 2, which enables the resistance to the anti-CD7 CAR T immunotherapy. So we use the alpha protein structure prediction and found because of this CD7 exon mutation, the antibody CD7 SCFV or antibodies cannot recognize this CD7 mutation protein that causes this CD7 negative relapse. Also, we found 2 patients relapsed with CD7 gene mutation, but in other 5 patients, we cannot find any CD7 gene mutation. So for these patients, we have the CD7 promoter methylation analysis, and we found after CD7 negative relapse, these leukemia cells are just in very high levels of methylation in the CD7 promoter. All these 5 patients have the same phenomenon. But when before CAR T therapy, their CD7 expression was high. This promoter was just in very low levels of the methylation. So we found just 2 cell lines. These cell lines have high expression of C7 expression, and other 2 cell lines have very low levels of CD7 expression. And we found for these high levels of CD7 expression, the CD7 promoter was in very low methylation level. But for these CD7 negative leukemia cells, they are in very high levels of the CD7 promoter methylation. So we try to use AZA, just demethylation agents, and we hope we can restore this CD7 expression. But after different concentration and different times of culture, we found that AZA can only partly restore CD7 expression. Maybe there are other mechanisms leading to CD7 negative relapse. So another complication is EBV-associated lymphoproliferative disease. We call the post-CAR T lymphoproliferative disease, just shortened as PCLD. So we just retrospectively analyzed our patients after CAR T treatment. 42 patients received CD7 CAR T therapy, and 6 patients had a detectable EBV DNA after infusion. And this include eight patients with initial EBV positive just before CAR T infusion, and 18 patients initially with EBV negative patients before CAR T infusion. For these 26 patients, 8 patients developed PCLD. And this is for 61 patients received BCMA CAR T therapy, and 58 patients received CD19 CAR T therapy. We found that none of the patients had a detectable EBV DNA after this CAR T cell infusion. So we think EBV activation is just a very special complication after CD7 CAR T therapy. Also, this is our 6 patients developed into the PCLD. We found that their prognosis is very poor. And this is for these patients, we just have this immunohistochemistry detection. So altogether, we have these PCLD samples for 6 patients. We also collected the PTLD 2 patients and EBV negative diffuse large B-cell lymphoma for 3 patients, and normal lymph node for 6. This as a control group and PCLD as our experiment group. Use these samples, we just have our single cell RNA sequence and special transcription analysis. So we found this CD7 negative, CD8 positive T cell dysfunction underlies the impaired antiviral immunity in PCLD, especially for this EBV virus. Also, we found the EBV is activated and induces malignant differentiation of the B cells. So this is my conclusion. Both donor-derived or universal CD7 CAR T cells achieve a high complete remission rate in relapsed refractory T-cell acute lymphocyte leukemia and CD7 positive acute myeloid leukemia. But easily, these patients are complicated with relapse, EBV infection, and pancytopenia. So our all-in-one strategy set CAR T combined with the allogeneic stem cell transplantation can overcome this CD7 positive relapse and pancytopenia. But CD7 negative relapse and EBV activation is still ongoing. So we hope someday we can find some strategies to conquer these CD7 negative relapse. And maybe some strategies can control this EBV infection. Okay, thank you.
Thank you very much, Professor Fu's wonderful talk about this CD7 CAR T therapy and your advances, the clinical practice and the basic research. So next we will end to the discussion section. And I think today Dr. Ye Yisan come from the Zhejiang University School of Medicine, First Avenue Hospital, and Professor Moti, and I think we will together, and we have this many wonderful experts, and we will join this discussion. Please,
okay,
Dr. Ye.
Thank you, Professor Huang. It's my great honor to coordinate this discussion session. I'm Dr. Yishan Ye from the First Affiliate Hospital, Zhejiang University School of Medicine. I think you have heard these exciting talks from our 3 top experts in CAR T cells. So first, I would like to propose to Professor Moti as a leading figure in the hematopoietic cell transplantation and cellular therapy in Europe, as well as across the globe. You have been deeply involved in the development and improvement of CAR T cells across the globe. So first, I would like to have your opinion or your insights on the 3 talks that is given today. And also today, this is a China European webinar based on IACH. I would like to also to know about your opinion or your perspective on the collaboration between China and Europe in the CAR T-cell immunotherapy.
Oh, thank you, Ishan, and thanks to all the distinguished colleagues who really delivered the beautiful talks. And I think the field of CAR T-cell is really moving rapidly, and we have to learn from each other because the experience can be different from one place to another. But one thing for sure is that the Chinese teams are moving very quickly in the development of CAR T cells. And I think this has to do with many factors, many parameters, but I think an important parameter is about the capacity of CAR T cells to allow, in principle, for a single shot treatment. Whatever the disease, the idea of using a CAR T cell is about trying to cure patient as a single shot or fixed duration treatment approach. And this is something where it seems to me that this culture is well established in China. We do have in many fields in the Western approach more delivering drugs over the long term approach. So I think this is an important aspect that we need to take into account, because these CAR T cells have the potential actually to cure many diseases. We heard about myeloma, of course, lymphoma is there, ALL, but now we heard already about T cell malignancies with the CD7 all-in-one approach. And I'm sure we'll discuss autoimmune diseases, although briefly, but clearly the spectrum is very wide, and we need more coordinated, and why not joining forces in running protocols.
Thank you, Professor Motti, for your insightful comments on the discussion today and your very important point of the one-step procedure trying to cure patients instead of these very complicated steps. So next, I would like to propose to Professor Huang as a leading figure in CAR T, one of the leading figures in CAR T immunotherapy in China. I would like to have your perspective on Chinese CAR T development, and maybe your vision and your opinion on the collaboration between China and Europe. Thanks.
I think it's very important how this is a joint meeting, and Professor Motti organizes IACH and Chinese Hematology Association with a joint meeting and focus on the cellular therapy. In the fact, the cellular therapy, CAR T therapy developed very fast in China and the Europeans. And today, I think the Europeans talk about this experience, CAR T therapy in European country. So I think in China, and more clinical trial and more commercial used treated different disease. Today, and we also invited Dr. Chen Zhu, and he is the first publication about the LLMP in vivo CAR T for SLE, the first paper. I think that is very important. So we can perspective in futures and more CAR T can be used as a different disease, including the hematological malignancies, autoimmune disease, solid tumor. And also, and we will cooperated the European countries, and so long time we have the many opportunities exchanging idea, acknowledge cooperation. So I think in future, and we have, I hope, this meeting and promoted all the cooperation in future, especially the young doctor and the young physicians involved in such the activity, just like the education process, and we will together the joint meeting, and we will have some joint together and cooperation project. So I hope the Professor Murthy as the leader of the WON, the hematology, and I hope we will have more the cooperation in future. Yeah.
Okay, thanks, Professor Huang, for this very important point, and also for IACH, not only for academic like today, but more for education and for the young physicians. So as you have mentioned that today we are also very delighted to have Dr. Chen Zhu. Professor Chen Zhu indeed is the director of the Department of Rheumatology from the Chinese University of Science and Technology. And we would like to also have your opinion, or what is your New England paper is about. We are very interested.
Thank you, Professor Ye, and actually, it's my great honor to have the opportunity to join this China-European webinar, which is related on the CAR T-cell therapy in hematological malignancies and autoimmune diseases. And so first, I would like to express my gratitude to the organizers of the webinar, Professor Huang and Professor Hu. So actually, I would like to say that as a rheumatologist, rheumatologists have already learned a lot from hematologist colleagues. Since you know that since 2021, with the first case of patients with SLE has been successfully treated by the autologous CD19 targeted CAR T cells. So this cellular therapy has actually ignited the passion of rheumatologists. So because before we did the cellular therapy, so we seldom see patients can get a remission in such a rapid time and such a deep remission. So that's why this cellular therapy has been developed very fast in the past few years. And actually, we can see the papers which published in the past 3 years, which is related on this cellular therapy on some systemic autoimmune diseases, including SLE, rheumatoid arthritis, and systemic sclerosis. So these indications so validated the efficacy of this CAR T-cell therapy. But however, we know that this conventional ex vivo CAR T has a lot of limitations. For example, the complex manufacturing, the high cost, which all of these aspects limited this broad application into the treatment of autoimmune diseases. So that's why the concept of in vivo has been proposed in the past few years. And since last year, actually, this strategy has been already moved from the preclinical stage into the preliminary first-in-humans clinical trial. And we can see that this lentivirus mediated in vivo strategy has already tried in the myeloma patients, which is led by Professor May. And our team has already performed the first-in-human study, which uses the lipid nanoparticle-based in vivo generation of CAR T cells in the treatment of SLE. So actually, compared with the lentivirus-based approach, this lipid nanoparticle-based approach actually has some advantages. For example, it does need to integrate the genomic DNA. So this is I think this is the most great advantages. But in other hands, so this advantage is a kind of a disadvantage. For example, so people may ask how durable will this CAR generation and the B-cell depletion. But however, we have already known that, and this is well recognized, that whether the CAR T therapy can achieve a durable remission rely on the whether you can achieve a deep depletion of B cells. So I think our rheumatologists have learned from the CAR T therapy is that if you can achieve a deep depletion of B cells in tissues, in the germinal centers, then it's possible, then you can achieve a durable or sustained remission. So that's why the expression time of CAR is not so important, but the depth of B cell depletion is the most important for achievement of a durable remission. So I think at the moment, the lipid nanoparticle-based approach also has some disadvantages. For example, it cannot have a long duration of CAR expression. But I think with the advancement of technical upgrade, it's possible to achieve a durable remission in the near future. For example, we have already optimized the strategy, and after we give the 3 to 5 doses every 2 or 3 days, we have seen already the positive signal that the B cell was depleted in the lymphoid tissues of the patients. So that's why I would say that it's possible that we can achieve a durable remission in the near future with the advancement of a technical upgrade. Thank you.
Thank you, Dr. Chen. I think you not only introduced your New England paper, but also you mentioned a very important point of nanoparticle mediated SMP mRNA and CAR T, which is the durability and and how to maintain the immune suppression, the CAR T cell efficacy, and to achieve the reset of the immune system, especially in SLE. So today we're also very glad to have Professor Li Chunrui, who is with us. I think she published a Nature Medicine paper on in vivo CAR T with lentiviral platform. So Professor Li, would you introduce us your study and your advantages in your study? Thank you.
Thank you, Chaz and the moderator, for a kind introduction and for organizing this important discussion. For our study, I think the most important message is that in vivo generation of CAR T cell can be done in patients with relapse and refractory myeloma, with early evidence of managed safety and anti-myeloma activity. There are 3 highlights I would like to mention. First, the treatment process was greatly simplified. ESOT was given as a single intravenous infusion, which avoids many steps of conventional extraviral CAR T participation. For many patients, long waiting time, complex production, and poor patient conditions are rarely clinical barriers. So the value of in vivo CAR T is that it may move CAR T therapy from a custom-made cell product toward a faster and more acceptable platform. Second, the treatment effect was encouraging. In this small phase 1 study, 4 of 5 patients achieved CR, achieved objective response, including 3 SCR. All evaluable response became MRD negative by day 60. Of course, the study was small, so this data should be interpreted carefully. Third, we observed an immune pattern different from competition CAR T therapy. There was a 2-phase immune response. The first wave occurs within 24 hours after infusion and was mainly related to innate immunity. The second wave occurred around days 6 to 2 weeks and was linked to CAR T expansion. I think this is an important biological finding and also a key point for future safety management. Looking forward, I think there are 3 important directions. First, we need to improve the vector and dose, aiming to reduce early innate immune activism while keeping effective CAR-T expression. Second, patient selection and risk assessment are very important. We should be especially careful in patients with high tumor burden, EMD, or lesions close to the spinal cord or central nervous system. Third, we need large multi-center studies and long follow-up to confirm how long response can last, long-term safety, the risk from gene insertion, and the risk of second cancers. Thank you.
Okay, thank you, Professor Li, for this very good introduction of initiative work and pioneering work. As we can see, the very good remission rate of your study in the phase 1 trial and the good safety profile. We are very looking forward to future phase 2 and phase 3 trials with this in vivo CAR-T with future developments. We are really looking forward. So apart from the CAR-T cells themselves, the microenvironment is also very important. And today we are also very glad to have Professor Florent Malard from the Professor of Hematology in Sorbonne University and Hospital Saint-Antoine. And Professor Malard is really a global expert, top expert in microbiome. And you released in a large EBMT meeting the very exciting results of the ARIES phase 3 trial on microbiota treatment for ruxolitinib and steroid refractory acute GVHD. So we would like to ask, because today is a CAR-T topic, so what's the future? I mean, how could the microbiome facilitate the CAR-T function and also maybe reduce the complications such as CRS?
So thank you, Ishan, for the great introduction. Thank you, Professor Wang, Professor Moti, for giving me the opportunity to discuss today to this great event with our colleagues from China. I'm very happy to be here. I would like first to congratulate the three speakers for the great presentation and for sharing this very exciting data. So, as you say, Ishan, we have a lot of exciting data in the microbiome field, mainly in the field of stem cell transplantations and for the treatment of graft-versus-host disease, when we demonstrate that the use of microbiotherapy is effective to treat patients that have graft-versus-host disease that failed by steroid and ruxolitinib. Nevertheless, besides the field of stem cell transplantations, over the last years, we have several publications that show that microbiome seems also to be involved in the setting of CAR-T cells. In fact, in those papers, they show that patients that receive some broad-spectrum antibiotics in the weeks before the CAR-T cells have a worse outcome with a lower progression-free survival and a lower overall survival. And when they look at the microbiome composition, they clearly show that we have a link between the microbiome composition and the patient outcome, where it will impact not only the complication, in particular the risk of cytokine release syndromes, but also the relapse risk. And very importantly, in the studies, they look more in detail because, of course, we may expect that patients that receive antibiotics before the CAR-T cell, this is because they receive more chemotherapy, have some more aggressive disease. And in fact, in some refined analysis, they were able to demonstrate that it was independent of the underlying disease severity, and it was clearly a link between the microbiome on the CAR-T cells, and in fact, it seems that the microbiome will impact the fitness of the CAR-T cells. So we clearly need to have some more data, to have more evidence, again, regarding the link between the microbiome on how we may be able to modulate the microbiome to improve the CAR-T cell fitness and, of course, the efficacy and maybe also the persistence of the CAR-T cells. On one we should investigate this is clearly if we can use a microbiotherapy as we are doing it in the field of hematopoietic cell transplantation on graft-versus-host disease to modulate the microbiome in patients that receive CAR T cell with some transplantation of the microbiome before the CAR T cell and maybe in the couple of weeks after the CAR T cells. And we already have some ongoing study in US, in Europe. So I hope that in the next 2 years, we'll have some first results to confirm what we expect based on the preliminary data.
Thank you, Florent. I think it's a very important point. I'm really looking forward to future data on how microbiome could facilitate or to help the CAR T cells to make it more functional. And we need also to see the insight to the mechanism behind. So in response to Professor Motti's important point on the one-shot policy and trying to cure the disease, today we have Professor Mingming Zhang, who is from our department, and he is focused on pushing the CAR T cells to frontline. And he published as the first author last year in JAMA Oncology about the Philadelphia positive ALL with dasatinib combined with CAR T cells showed very good results. So what is your point? I mean, how can you introduce your study? And also, what's the future of putting the CAR T cells to frontline, Professor Zhang?
Okay, thanks, Yishan. Yes, CAR T cell therapy, of course, have achieved very good efficacy in relapse or refractory patients with hematologic malignancies. But we found that patients with heavily pretreatment, that patients' T cell, not only the decreased T cell count, but also the impaired T cell function that makes the CAR T cell therapy impaired efficacy. So we wonder if we can move the CAR T cell to the first line to when patients have a very good T cell function to receive the CAR T cell therapy, and then even to substitute like chemo or transplantation. So at the early after 2021, we begin the clinical trial of the first-line CAR T cell therapy for newly diagnosed hematological malignancies. First of all, we bring out the CAR T cell first-line therapy for Ph-positive ALL because the Blincyto have established its efficacy in the first-line therapy of these patients. And at that time, we are not that confident, so we bring out the sequential CD19 and CD22 CAR T cells for these patients. But now we found that only one shot of the CD19 CAR T cell therapy is enough, and most of the patients in our study, 92% of patients will have the disease-free even after a follow-up of median until now 3 years, and without transplantation, without chemo. And now we also finished the first-line CAR T cell therapy in high-risk large B-cell lymphoma. Not like the ZUMA-12, we combined the ZR2 that without chemo. 2 cycles of ZR2 combined with first-line CD19 CAR T cell therapy, and we found the CR rate after CD19 CAR T cell therapy is very high. I can't remember exactly, 95% of CR rate, and only 4 patients relapse out of the 40 patients. So also, I think the first-line CAR T cell therapy in high-risk large B-cell therapy makes patients can cure without chemo or without auto transplantation. So I think we are now designed to the phase 3 trial or phase 2 trial to compare first-line CAR T cell therapy with the traditional regimen to see the advantage of the first-line CAR T cell therapy. And thank you.
Thank you, Professor Zhang. I think that's very important strategy and moving forward for CAR T cells to frontline. and we are very optimistic about it. So today we have Professor Meng Lü, and he told me that he would like to study from the slides of these experts, and maybe you would like to propose some questions, or what's your opinion? Because I know that you are doing a lot of clinical trials on T-cell malignancies, and it is very challenging. Today we have Professor Hu and also many experts. So maybe you can focus on this point and show your opinions and your perspectives. Thanks.
Thanks, Yishan, in introducing, and thanks Professor Murty, Professor Huang for this excellent seminar. And as we all know, the T-cell malignancy is not a good task, and we have solved this fibrosis problem, and we have also solved the ACAT problem post T-cell lymphoblastic leukemia. And in our Peking University, if the patient have the same tumor burden, if it's T-cell leukemia or B-cell leukemia, and they will have the similar ACAT incidence, just related to the tumor burden, not related to the CAR-T type. So we have the confidence that our patient would not need to receive any transplantation within 3 months post the CD7 CAR-T therapy. That's the base that we can make this product be approved by the Chinese FDA, that's MPA. And the second question, I'm very interested in Professor Hu Yongxian's work on the EBV PTLD, because we currently are focusing on the CD5 CAR-T. And CD5 CAR-T have the problem of the elevated virus reactivation compared to CD7 CAR-T, especially the EBV reactivation and the CMV reactivation. We are still trying to focus on what's the mechanism behind this disparity. Professor Pan Jing has published their data addressing the difference between the CD5 and CD7 CAR-T in Advanced Science this year, but we think it's maybe not the whole picture that need cooperation between the Zhejiang University, our European friends, and Peking University to solve this problem. Once the CD5 can solve the EBV reactivation problem, maybe it will be the next CD7 CAR-T all available to the world. Thank you. And maybe Professor Hu, maybe you can address what my comments. Thank you.
Professor Hu. I think maybe Professor Hu is. Can you answer the question? Maybe CD5 can replace a CD7 or become another CD7 if PTLD or PCLD is overcome?
Hello, Professor Lü. Can you repeat again?
The simple question, and we do a lot of CD5 CAR-Ts nowadays, and we found elevated CMV and EBV reactivation is especially EBV PTLD following CD5 CAR-T. So when we try to make the success following the CD7 story, we must to solve this problem, the EBV reactivation post CD5. Now, what's your opinion? Because you have done a lot of excellent work addressing the mechanism of EBV reactivation. Thank you.
Okay, very good question. Do you have as a enrollment criteria for the EBV DNA positive patients? Will you exclude it or include
no.
it for this patient?
If the patient had the EBV reactivation before the CAR-T, actually we will not get the patient get infused. We will exclude this patient. But and 90% of Chinese people will have CMV IgG positive. So we could not rule out all these seropositive patients. We just rule out the active EBV DNA reactivation before CAR-T.
Yes, but for our CD7 CAR-T, we do not find as a CMV activation much higher. Only the EBV activation, I think is a bigger challenge. So I think
that's why we want to maybe we need a cooperation in near future to see what's the difference between the CD5 and the CD7 CAR-T. Maybe the mechanism is totally different from CD7.
Yeah, because in my opinion, if we knock out the CD5, the T cell function will be enhanced.
Yeah.
So,
yeah, sure.
So maybe there are different pathway for this anti-EBV immunity.
Yeah, at the very beginning, we think maybe the CD5 CAR-T will irradiate all the CD5 positive B cells, so you will get an immune deficiency both in T cell and B cell. But our hypothesis has been proved wrong. The B cell has little effect about the B cell. It's just the impairment of the T cell function. Maybe, but we didn't find the solution. So
yes, I think
because you have done a lot of the single cell analysis, do you find any clues that which patient will be the victim of EBV PTLD?
I think this is
feature?
the Dr. Meng. This is really a fascinating topic, and obviously, what I can hear from this discussion is that we clearly need to have a special webinar dedicated to CAR T cells and T cell malignancies. We need to have a special, and we will discuss all of these issues. We need to have a webinar dedicated to autoimmune diseases, but also to the difficulties also that can arise and challenges with in vivo CARs. But we are already running very late, so I'll hand it again to Dr. Ye for the last few questions before the conclusion.
Okay, thank you, Professor Moti. We have I forgot to mention that Professor Liu is from the Peking University People's Hospital, a young expert in CAR T cells. So we have the last panelist is Dr. Chen Jia, who is from the Suzhou University First Affiliate Hospital. And I know you are leading several trials on AML, and AML is also a big challenge for CAR T cells. So maybe your opinion or your perspective on the CAR T cell in myeloid diseases, or you have any question for the speakers. Thank you.
Thank you very much for the introduction, and thank you very much for the kind invitation from Professor Huang and Professor Moti. So certainly, I think today we have talked about the myeloma, the T cell lymphoblastic leukemia or lymphoma, and talked about the in vivo CAR T. But I think the acute myeloid leukemia takes the majority proportion of the acute leukemia patients. So maybe we should find some way out for these kinds of patients, especially some of these patients are very elderly and fragile and cannot tolerate high dose transplantation or high dose chemotherapy. So that's why I think we should pay more attention on CAR T development in this kind of patients. So I think CAR disease where relapse remains common and CAR T has revolutionized the B cell malignancies, but AML patients the unique hurdles. I'd like to talk about several aspects about the AML. I think the first one is how to choose the right target. I think it's Achilles heel for AML patients, and ideal antigens are uniformly expressed on leukemia cells, but absent for healthy hematopoietic assays. So in reality, most of AML targets used now, such as CD33 or CD123 or CL1, also appear on normal myeloid progenitors, cause our target of tumor toxicity and maybe prolonged cytopenias. So we also trying to develop some dual targeting CAR T cells, including CD38, CL1, or CD33, CD123, or logic-gated CAR T cells with and or not gates to spare normal cells. But I think the antigen escape is a very big problem for myeloid patients because there is a very diverse subclones of myeloid leukemia, and the pathogenesis of myeloid leukemia always involves several hits, which may lead to the immune escape from the myeloid patients. And I think there are some efforts on the car structures optimizing on the myeloid battlefield. The conventional second generation cars often fail in AML due to maybe the chronic stimulation by low-level normal antigens. But third generation, like truck cars, which secreting IL-12 or IL-18 or switchable cars, may contribute to this kind of obstacles. But the tonic signaling will be a challenge in future development. So maybe we can try some CAR NK or CAR macrophage, just mentioned by Professor Mehan, with different activation thresholds may optimize this kind of problem. And we have talked about a lot about the microenvironment, especially by Professor Malach. I think there's also the problem of leukemia microenvironment in myeloid patients. And the AML bone marrow, I think, is a hostile circuitry with MDSCs or Tregs or hypoglycemia, and especially, particularly the TGF-beta. Cars with dominant negative TGF-beta receptor or the chemokine secretion may overcome part of these questions, but we also have some physical barriers. So maybe we can do some combination with other drugs to destroy the negative microenvironment, or radiology therapy may to degrade the extracellular matrix. And I think there's also some rationale combinations to break resistance, which, for example, with BCL2 inhibitor or some hypermethylating agents to destroy the negative environments. And then there are some comments which I get derived from the discussion tonight. And one is the genetic engineering to enhance the myeloid tumor recombinability, because we know that there's no tumor-specific antigen, especially for the myeloid patients. And beyond engineering T cells, maybe we can try some method to modify the tumor site. Many AML cells downregulate the surface antigens and co-stimulate molecules, and I think it's a form of immune invasion. Maybe we can force antigen expression using CRISPR or other activated methods or viral vectors to upgrade the safe immunological surface proteins such as B7 or CD70 or synthetic sensor antigens, IM-blood, but maybe it should still in the laboratory, and we can do some maybe epigenetic reprogramming via histone deacetylase inhibitors or other drugs. I think it's also a kind of direction we can use. And there another point is about in vivo CAR. In vivo CAR generation delivery is also, I think, promising in myeloid patients. And Dr. Mehan has introduced the different kinds of the vectors or carriers and the advantages and the limitations. So there's one question for Professor Mehan, if you are still here. So maybe I have ignored, if you have evaluate the in vivo CAR, the carrier of in vivo CAR to different kinds of immune cells. For example, you have evaluated different subtypes of T cells, and I'd like to know what happened to other immune cells such as NK cells or macrophages or even the neutrophils. And when the in vivo CAR virus infected these kind of cells, will it be eradicated, or you also will transfer these immune cells into CAR cells?
Okay, thanks for Professor Chen's very profound question, but I think it's very difficult to answer now because there are many translational studies for in vivo immune cells, not only for T cells, but also for NK cells and the macrophage now. For my opinion, I tried with in vivo NK and the macrophage too. Usually we use the CD7 to both at the CD7 for T cells and for NK cells. But for NK cells, as we know, they are not from the adaptive T cells. So when you transfect it for the NK cells, the expression is very difficult, and also the activation is not as much as T cells. So I think only for NK cells is not enough for the functional work in the de novo tumors. And for macrophage, it's also a big problem because for macrophage, they have different, at least two types. That's the one is Th1 and the Th2. For the if with the two type, maybe there is more phenotyping for inhibition. So we should how to make it from 2 to 1. That is also very important. So we try to use some cytokines together to do this activation. So I think that is also a very important way. But all of these are in the preclinical study now, not in the translational. So, but that's a direction in the future. So not only for T cells, but in vivo immune cells is a new strategy in the future. Thank you.
Thank you very much.
Thank you, Professor Chen. Thank you, Professor May. It's a very exciting discussion. So time flies. Let's welcome Professor Moti to give us very fruitful, webinar to give us an closing remark. Thanks.
Thank you, Ishan, and thanks to all the colleagues who contributed presenting or in the panel discussion. I think this first joint ICH Chinese Society of Hematology webinar has taken us from the origins of CAR T cells to its transformative present, but also very, very promising future. As I have just mentioned a few minutes ago, I think it's about setting the stage, and we definitely need to have more focused webinars and discussions on more narrow topics, because today we have discussed the broader picture, but I'm sure we will have some focus on more specific topics. And this marks the beginning of what we hope will be a long and fruitful collaboration, true again to our spirit at the ICH of Hematology Without Borders. We want science, innovation, but also patient care to transcend geography. And clearly, we have achieved and reached this goal thanks to all of you, but also thanks to the support of our leaders, Professor He Huang and Professor Yu Hu. Maybe you would like to add a word, Professor Hu?
Dear colleagues, thank you for joining this wonderful symposium on CAR T cell therapy. We have held several excellent presentations from Professor Matthias, May, and Hu on European CAR T practice, in vivo CAR T, and CD7 targeted CAR T therapy. The panel discussing fostered valuable Sino-European exchange. I hope everyone gained new insights and inspiration. The Chinese Society of Hematology and IACH will continue to organize a series of meetings on another fields, such as transplantation, leukemia, to consolidate the cooperation between the European and China. Thank you to my co-chair, Professor Moti, Professor Huang, all speakers, and panelists, and the audience. Good night.
Okay. Thank you.
Bye-bye.
Thank you.
Thank you all.
Thank you.
Thank you. It's a wonderful meeting.
Goodbye.
Thank you. Thank you very much.